Good morning. We're live. And how's everybody doing? Whatever. So I left the exams out on the mantle next to a plate of cookies, hoping that the elves would grade them last night. And I woke up this morning and the cookies were gone. And Quinkle, our dog, didn't seem to know what happened, but the exams haven't been graded yet. They'll get graded. All right. And just so you know, we didn't have lab yesterday. We're not having lab tomorrow. Whatever. All right. So we are kind of on the last lap here, or instead of laps, last third, talking about some old-fashioned cell biology, some mechanisms of protein processing and vesicular trafficking and things today, and some interesting stuff. So, let's begin. The question arises, how do proteins know where to go? You know, our primitive cell biologist, faced with cells, we've begun to figure out, about the phospholipid bilayer. And we know there are proteins in the phospholipid bilayer. How do they know how to get in there? We know that certain cells are going to be secretory cells. And a really good experiment that was done probably, and as I was thinking about the experiment, I'm not sure who did this, but it had to have been done after World War II because it requires a bit of radioactive tracer stuff. And in fact, a couple of the things we're going to be talking about really only could be done because of radioactive tracers. Radioactive tracers aren't used so much these days. We've got microscopy using antibodies and fluorescent molecules. We've got a range of different tools. But if you want to track a protein, one of the best ways you can do is tag it with a radioactive tracer. Specifically, you can purchase and you can have made for you a sulfur 35, or S35 as it's called, methionine, is a really good radioactive tracer for looking at proteins, because proteins are the only polymers they're going to pick up S35 methionine. So you can treat cells with radioactive sulfate or with radioactive nitrogen and things. And in fact, in order to make S35 methionine, that's what you do. But what you want to do here with a labeling experiment is something that's going to be specifically taken up by the system you're interested in. So S35 methionine works pretty well. And so what we're going to do is something in the biz known as a pulse chase experiment, in which we're going to take some secretory cells, like cells from a mammary gland of some kind, something that secretes a lot of protein. And we're going to put it into some media, hit it with a very large amount of S35 S35 methionine for about 15 minutes. That's going to be the pulse. And then after letting the cells sit in S35 methionine for 15 minutes, we're going to wash the media real well, wash the cells, and replate them, and now begin watching where that pulse of radioactivity goes into the cell. It's a little bit like if you wanted to study an escalator, because you're saying, wow, there are steps that are magically appearing here, and then they disappear up there. I have a hypothesis. They're being eaten and then constructed. And one of the things you might want to do is track the movement of these stairs. And so a great way to do this would be to take a can of spray paint, and as the escalator is moving, you say, okay, for a count of 3, I'm going to spray red spray paint. You're spraying this patch. That's the pulse. And now you watch it roll through. That's the chase. So you're chasing it into the system. And if you've never seen an escalator, I can tell you these are stairs that magically move. Everybody knows an escalator. Just want to go on record, I don't know. I'm trying to think of the last time I rode an escalator. And once you do this, you watch and you see that that red patch of steps is moving up. Okay, I expected that. It goes up to the giant eater, it disappears, and it's like, okay, it's gone. And then a minute later, here comes your red patch of cells. And you now have a much better model for how this magic staircase works. So a pulse trace experiment gives you an understanding of a dynamic process that is going from, starting at time 0 and moving its way through to time 15 or something. So what we're going to do is every 30 minutes, why not? We're going to take a few of the cells, and a standard experiment would be to extract the proteins and look at them on a SDS gel by size. But what these guys did is something that is called autoradiography, in which, and as I think about this, it is a critical experiment, but it's quaint in its technology. You do know that before the invention of video cameras and video imagery, that pictures were taken on a material called film. And black and white film is, in fact, a sheet of plastic coated with gelatin embedded with silver grains. And the silver grains, when they are hit by either light, a photon, or a radioactive particle, they oxidize and they change their chemical state. And then you put them into a developer, which will render the non-oxidized, or regardless, the non-hit silver grains soluble. They wash off, and the silver grains that got hit by a photon or a particle of radiation, they are insoluble, and they end up as a little black dot. And you create a photographic image this way. So, people I don't think even film is even sold anymore, but that was a critical thing. And what you're doing is taking the cells, putting them onto a glass slide, and you can observe them under a microscope, but taking that glass slide and dunking it into photographic emulsion. So they used to sell photographic emulsion, and it's in gelatin, so you have to warm it up, and you got to do this all in the dark. And you take the slide and physically dunk it in the photographic emulsion and hang it up to dry, and you let it expose for a couple of days. Now the cells are dead, so wherever the radioactive material is, it's fixed, it's stuck in that spot. Well, this is called autoradiography, and that's an A, autoradiography. And what you begin to see is this. At say time 30, we see in the cell dots of radioactivity surrounding the endoplasmic reticulum. You're going to see dots of radioactivity in lots of places, but you begin to see it accumulate first in the ER. Specifically, what you would know as the rough ER. But as we're about to see, endoplasmic reticulum is just endoplasmic reticulum. It may have ribosomes on it, may not, but it's all endoplasmic reticulum. So the rough ER starts to show the label. Now, as we go to 1 hour, we begin to see the dots. So here's our nucleus, you see, and they are no longer in the ER. They actually are starting to get into this stack of material that we call the Golgi apparatus, which is a stack of membranes. We'll look at it actually in a lot more detail in a few moments. And we start to see the radioactivity. Because we have multiple markers, we'll draw the radioactivity in red. So here's our radioactivity in the ER. The Golgi is sitting here, it's cold. Now the radioactivity is in the Golgi, and the ER is cold. Because this is a chase. What we've done is hit that little spot of spray paint of radioactivity, moved it through. And next, we'll go ahead and jump forward 2 hours. We see now that the protein is, I'm carried away with my animation, but it's worth the point. No longer in the ER, no longer in the Golgi, but in fact now it's in little secretory vesicles. And about 3 hours later, the radioactivity is not in the cell at all. The radioactivity is now floating about in the media. We have tracked protein through a secretory cycle. This is a pretty cool trick. And it begins to suggest that proteins are making their way through the ER, the rough ER, into the Golgi, and from the Golgi into secretory vesicles, and then making their way out. So fairly quickly, 50s or so, we begin to understand that there is a secretory process and that some proteins have the ability to actually make their way through in this pathway. So if we begin to think about this from a, just sort of a reflection perspective, there are compartments in the cell. And we can begin asking now the question, okay, so this is, we see proteins are being secreted. They're going through the ER. What are some of the other compartments we might think about, and how are proteins getting into those spots? It's going to require a little bit more technical expertise, because now rather than having to just label this thing with a generic S35-methionine, we're going to have to track individual proteins, but we can do that. And so we've got some ideas of compartments in the cell. We've got, of course, the cytoplasm. We have proteins on the cell surface. They might be embedded in the membrane. They might be peripheral, regardless. We've got secreted proteins that are being dumped out into the extracellular space or into the media. We've got proteins that are inside the various organelles. That is, we have mitochondrial proteins. We've been talking about a lot of those. Another good compartment to think about is there are proteins that go back into the nucleus, and they're actually carrying out things like RNA polymerization and transcription factors and stuff. So we know that proteins have got to have specific compartments that they reside in. And the question becomes, how do these proteins get there? That is, protein targeting. What is the mechanism? And it turns out it's not a single mechanism. There are a number of different mechanisms that are critical for proteins moving into and out of the cell and finding themselves in different specific locations. Let's just take, as an example, nuclear localization. To get into the nucleus, it's actually a very easy thing to do, and you can observe this in native proteins. You can also, using recombinant DNA technology, force a protein to go into the nucleus, which is actually can be a very cool little trick. Rather than having your protein, your green fluorescent protein, being spread throughout the cytoplasm, you can have a nuclear localization signal that will target it to the nucleus, and you'll have this nice, crisp little green dot. So nuclear localization is a pretty cool trick. Turns out, for nuclear localization, there is something known as a nuclear localization signal. This is an amino acid sequence that's actually part of the protein, an intrinsic part of the protein. And it is essentially the signature that says, yo, I belong in the nucleus. It ends up actually being a stretch of lysines. It is pretty much just a stretch of basic or positively charged amino acids usually at the amino terminus, which would correspond to the 5' end of the RNA. That is sufficient to target a protein into the nucleus. Now, this is actually happening after the protein has been translated. So, just as a reminder, protein translation occurs in the cytoplasm in eukaryotes. Prokaryotes, they got a whole different kind of game. But in the eukaryote, the RNA has been transcribed. It is taken out into the cytoplasm. The ribosome binds the 5' cap, sets up shop at the AUG, and you begin making protein. So the protein has been translated. Once it's been translated, it has this nuclear localization signal hanging on, and there are systems that pick that up and target it into the nucleus. This would be known as post-translational translocation, which is a delightfully alliterative term. Translocation. Post-translational translocation is a term we use when proteins are translated in the cytoplasm and then they're targeted somewhere. And just as a little point, post-translation translocation is how you get stuff into the nucleus. It's also how you get stuff into the mitochondria. Turns out there are signals, there are amino acid signatures that will put something into the inner membrane or the outer membrane. There are amino acid signals that will put stuff into the nucleus. There are amino acid signals, if you're a plant, that will put stuff into the chloroplast. So we've got then a protein being translated, or an RNA being translated. The protein is translated, and if it doesn't have these signals, it's going to hang in the cytoplasm. That's where proteins belong. You know, all these different glycolysis enzymes, all these different structural proteins we've alluded to. All kinds of stuff is happening. They are in the cytoplasm. If you've got a sequence that says, hey, I belong in the nucleus, well, you get ushered into the nucleus. All post-translationally. All right. The next step was figuring out, or I guess we can kind of think about this, cytoplasmic means you just ain't got no signal. Organelles, nucleus, post-translational translocation. Now, to get things into the membrane, this outer membrane, and to get things secreted, that is a lot more complicated. And it takes a while to begin figuring this out. One of the first things that was done, and it was actually a very early recombinant DNA experiment, and so this is stuff being done back in the late 70s, early 80s, by two fellows, Gunther Global and his buddy, maybe Hans, I can't think of this guy's first name, Doberstein. Global and Doberstein, they deserve a shout out. I am sure that both of them are no longer on this terrestrial plane. I did have the opportunity to meet Gunther Global once. He was about a 7-foot tall guy, as I recall, and he had hair that stood straight up like this, with a little bit like a racer head. A fascinating gentleman. I don't think he won the Nobel Prize at that point. I'm not sure he won the Nobel Prize. He presented a seminar, an hour-long seminar, and he had like 3 slides. And you might think, wow, this guy was so engaging that he could present an hour-long seminar with just 3 slides. But no, he actually was a pretty boring seminar. Sorry, Gunther, but it really wasn't all that good. He is no longer with us, though. Regardless, working out of the Rockefeller Institute in New York, Global and Doberstein published a series of papers in which they began to elaborate this process. And their first experiment was this. They had 2 different proteins that they identified. Proteins that were really easy to clone, like, The serum albumin protein would be a good one to do. We were using this as part of our protein assay system. There is a major protein found in blood serum of all animals that have blood. And from cows, bovine serum albumin is really easy to get because cows got lots of blood. You can precipitate this stuff with cold ethanol after you split the red blood cells out, and you can make purified BSA. So bovine serum albumin is secreted. Now, what we're going to do is look to see what's happening and why a secreted protein is secreted and why a protein that belongs in the cytoplasm isn't secreted. So pick an enzyme in the glycolysis pathway, any enzyme. There are a lot of cytoplasmic proteins that would be easy. You could pick the globin gene is another one that would be like massively amounts of it that's present in the cytoplasm of red blood cells. Regardless, we're going to do the experiment like this. Now, what Blobel and Doberstein are employing is a couple of tricks. Rather than doing autoradiography, they're taking advantage of a system that was probably coming back in the 40s and 50s, and it's one of the ways that the genetic code was figured out. And it's something called in vitro translation. And so in vitro translation is still quite the thing. In vitro translation, as the name implies, is translation carried out in vitro, meaning it is a cell-free translation system. And you can make these, you got to start with a cell, but there are a couple of different translation systems. One uses red blood cells from little bunny rabbits known as a rabbit reticulocyte lysate, where you take the red blood cells and you gently lyse them. And this lysate now is in a little tube. It contains all of the things you need for translation. The ribosomes are there, the tRNAs are there, you have ATP there. You can degrade the RNA that was present, the messenger RNA specifically, and then you can put your RNA, essentially you're programming this in vitro translation mix. And in order to make this thing really sing, you also throw in, again, a radioactive tracer, S35-methionine. And typically you run this out in a gel. So now, if we imagine we take a red blood cell and lyse it, there is a ton of protein in that little tube. All the proteins are in that red blood cell. But only the newly translated protein will be picking up our S35-methionine. So consequently, if we label it with S35-methionine, we can look for a new band by now, again, autoradiography, but instead of using cells, we're going to have a gel, a protein gel, that separates out proteins based on size, grind that gel down, slap it on a sheet of x-ray film, let it sit in the refrigerator for a couple of days, and then you develop that film. And so when you're looking at an autorad, you have the actual gel. A lane on the gel might look like this, with just a ton of proteins, and some in very small amounts and some in huge amounts. But when you put that on a sheet of x-ray film, you're going to see beep, 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 a single protein is labeled. Can you see that? Yeah, you can see that. All right, so by doing in vitro translation followed by autoradiography, you can keep track of this protein. You can watch it get translated. So this is what Blobel and Doberstein did, is they chose two different RNAs, an RNA that would produce a secreted protein and an RNA that produces a cytoplasmic protein. And they ran a lot of gels. And what they found was this. So if we've got the secreted protein, which we'll label with S, versus the cytoplasmic protein. First off, what we're going to do is look at these two proteins in the native cell. So we have immunological ways of doing this, all kinds of ways, but just for the sake of our diagram, this is what we got. We have a secreted protein, and it'll be right there. There's the band at some specific size. we have the cytoplasmic protein. And it'll be right here at some specific size. Okay. Next, they take the RNAs, they do in vitro translation. So this is, if you want to get all Latin-y, this would be in vivo, that is, within the cell. And now we're going to do the in vitro. We've got the secreted RNA, or the secreted protein, the cytoplasmic protein. And now when they do this, lo and behold, they see an interesting little change. First off, the cellular protein, or the cytoplasmic protein, is exactly where it was. Same exact spot. But now, the secreted protein is a lot bigger. It went from being this size, to that size. That's odd. And I can well imagine that Blobel told Doberstein, dude, you screwed something up, run it again. And he comes back with the same result. It's like the secreted protein is larger during in vitro translation. And Blobel was like, I don't believe it. Do it again. And after the third or fourth time, Doberstein came up to him and said, man, it is bigger. I don't know what to tell you. And Blobel went, oh, okay. So he goes back to his office and thinks for a while. What they next do is an experiment in which you can take the vesicles from a cell. You can purify vesicles from a cell. And it's typically vesicles purified from a dog pancreas. Why they use dog pancreas vesicles, I don't know. But that's what is commercially available. If you do in vitro translation, now, with the dog pancreas cells, and so we've got the vesicles from the dog pancreas. So we add the vesicles, and we look at the secreted versus the cytoplasmic. This is what Doberstein was able to report. He comes back and says, boss, check this out. The cellular cytoplasmic gene is, once again, in its same spot. And now, the secreted protein is back at its lower spot. And Blobel's like, that is interesting. Let's try this experiment. And Doberstein was like, what, boss? Blobel says, if the vesicle is taking the protein up, it's not going to be sensitive to a protease. And so if we do in vitro translation with the vesicles and throw a little bit of protease into the mix to break down the protein, and then you run the gel, what's going to happen? And Doberstein's like, there's not going to be any protein left. But Blobel, he's smart. And when they run this gel, right room here for my third or my fourth gel, this is what they see. So now we've got our secreted versus our cytoplasmic. We're doing, again, in vitro plus the dog pancreas vesicles. And now we're going to treat it with a protease after we've done the in vitro translation. And here is what Mr. Doberstein saw. He comes back and says, boss, the cytoplasmic, he says, boss, I don't know if I screwed something up. The cytoplasmic one is just barely there. There's hardly anything there. There's a big blob of radiation down here. But the secreted one is still here, still strong. It's right where it's supposed to be. And Blobel was like, I knew it. And Doberstein's going, you knew what? You didn't know jack. No. What had happened is the secreted protein ends up inside the vesicle. And so it's not sensitive to the protease anymore. and the cytoplasmic one gets broken down. What we have here is a mechanism by which a protein can get put into a vesicle. Now, one of the things that we're going to have to figure out is this size change. In the absence of vesicles, the secreted protein appears larger. So they actually published multiple papers on this, and from like 1975 to 1985 or so, they're working on this. And it's Blobel and Doberstein, Blobel and Doberstein, Blobel and Doberstein. And what they began to figure out is, in fact, a grand model for how proteins are being placed into vesicles. And it goes like this. One of the things they start doing is purifying the vesicles and looking to see how they're interacting with that RNA and with the in vitro translation protein. And they come up with this model. The secreted protein has an amino acid sequence on it that Blobel and Doberstein call the signal sequence. And the signal sequence is, in fact, fairly conserved. I mean, you can actually do a blast search, the way we did for our amino acid sequences. You can do a blast search on all of the proteins in the database, and you can find proteins that have a signal sequence. These are proteins that are going to be put into vesicles or be secreted. So it's a fairly specific signal. And the way this works is the following. When they took vesicles and they began to wash them with a moderate salt solution, they could knock off a protein from the vesicles that was allowing the signal sequence to get taken up into the vesicle. And so they determined that the vesicles themselves have a protein on their surface that is interacting with the signal sequence. Further, they began to play around and they identified something that they called the signal recognition particle. The signal recognition particle, which Blobel and Doberstein aptly named or abbreviated as the SRP. And once they purified it, they started breaking it apart. The signal recognition particle actually is a ribonuclear protein complex. It's got a little small RNA, and then there are a set of proteins on the SRP itself that are interacting. And they do a couple of things. The SRP, number one, binds the signal sequence. So as the RNA is being translated, if a signal sequence comes out, SRP binds it. If the protein doesn't have a signal sequence, it's just translated and it hangs in the cytoplasm. But if it has a signal sequence, SRP binds it. And the second thing that SRP does is it's going to halt further translation. Halt. So we're going to halt translation after SRP binds a signal sequence. The third thing it does, and it's probably diffusion, but regardless, it now is going to dock onto the ER. And it's interacting with a protein complex on the ER known as, and not all that creative a term, docking protein, also known as the SRP receptor. So now, what we have got here is the mechanism by which plain old ER becomes rough ER. Because we've got a system that's looking like this. And we're going to take a moment and throw up some, maybe just throw up, but put some pictures up from the book to demonstrate this. So we've got an RNA hanging out. There's the poly A tail. The 5' cap is here. The small subunit binds, moves down till it finds the AUG in the appropriate context, and we begin translating the protein. So the protein's coming out. And if this protein actually has the amino acids that make up the signal sequence, SRP will bind. So here comes SRP. And SRP, once it binds, it halts translation. This whole mix now is going to bind to docking protein, the SRP receptor, on the ER. So let's load it onto the ER. Here is the grand endoplasmic reticulum. This is this giant membrane structure in the cytoplasm. And here is the SRP receptor. Now, when SRP binds to its receptor, translation is initiated again. But this time, and we're going to sort of redraw the whole shebang here. We got the surface of the ER. We have the SRP receptor here, kind of looking like a blob, and then there's actually a little bit of a hole in here. And the signal sequence is going to hang right here. And the protein is now being dumped in through this hole as it's being translated. All right, what we got here is very much like a long piece of spaghetti, and you have one end of it that is the signal sequence. You grab it, and then you begin dropping the spaghetti through the hole next to it. And this is the protein moving into the ER as it's being translated. And so Blobel and Doberstein call this not post-translational translocation, but this is co-translational translocation. So Blobel and Doberstein and the co-translational translocation system. It's almost as good as Harry Potter and the Goblet of Fire. All right, so this is how proteins are getting into the ER. And the reason I say this is what makes rough ER rough is the ribosome ends up hanging right here, pumping the protein in through this little tube. And so you end up with a bunch of ribosomes sitting on top of the ER, pumping their proteins in. And this is a key point. We're going to elaborate on this on Friday, but let's look at a couple of figures. They may be worth them 1,000 words that I just spent the past 40 minutes on. So it's a little bit of a story, but it's not too hard. And you do need to kind of just look at some diagrams and think about it a bit to really fully get onto it. But let's see if we can. make it a bit easier to see. Chapter 15 is where we are. Bing, 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 bing. So, this is an electron micrograph of the cytoplasm of a secretory cell. And ain't that packed with stuff? You know, it's one of the things that we don't typically think about, but the cytoplasm in a cell is like the consistency of Jell-O. It is packed with protein. Proteins are at such a high concentration in the cell that I don't think you can generate that level of protein concentration outside of the cell. I mean, it is incredibly viscous, really interesting mix in here. There's a lot of surfaces, there's a lot of stuff happening. So we've got these dots that are just cytoplasm. Here's some vesicles. There's a mitochondria. Actually, that's a mitochondria there too. There are vesicles that have these different terms, things like peroxisomes. Here is some rough ER, and you may not be able to see on the picture, but it's just a whole bunch of little pancakes, little slices that would be sliced across it. And this is a bit more of a cartoony figure, but there you go. So we've got the endoplasmic reticulum with the membrane-bound polyribosomes, ribosomes that are interacting with the membrane. There's the Golgi apparatus here, and then we have a whole set of different secretory vesicles coming out and fusing with the cell surface. So also we have down here free ribosomes. All right, so just a little list here of some different kinds of compartments. Yeah, we're covering it, whatever. And do you really need to memorize the relative volume and number of the major membrane-enclosed organelles in a liver cell? Now they say that there are 1,700 mitochondria in a single liver cell. That seems phenomenally high. But if they say it, it must be true. Okay, fine. Yeah, these membranes were engulfed at some point, but that's not the point. All right, here are some signal sequences. We have the standard actual Blobel and Doberstein signal sequence that imports the protein to the ER. The thymine, serine, phenylalanine, valine, serine, val, gly, isoleucine, lu, phi, trip, ala. Go ahead and memorize that. I'll ask you that. No. What you can do, though, is if you attach that to your protein, it'll get targeted into the Golgi, into the ER. If you want something to stay in the ER, there's actually a ER retention signal. You want something to get imported in the mitochondria, we got this, imported in the nucleus. And just as a little note, nucleus and mitochondria, these are post-translational. After the protein has been translated, they get picked up, not co-translational. So, this then is one of Blobel and Doberstein's experiments, where they took the signal sequence, the first 20 or so amino acids, off of a protein that they know is in the ER, and they actually attached it to a cytosolic, to an RNA that produces a cytosolic protein, and carry out the cell operations, and you end up then with the cytoplasmic protein now stuck in the ER, and the normally ER protein, if the signal sequence isn't there, it remains in the cytoplasm. And that's actually a critical point we'll get to in a moment. Yeah, the nuclear envelope, we'll talk about later. Nuclear pores, these are what, this is the space where things are being brought in. And there's a whole set of things that are carrying out nuclear importation. So you have the nuclear localization signal on the protein. The protein has been translated, so it's post-translational. The nuclear localization signal interacts with some kind of importer, and it gets pulled into the nucleus. The same kind of thing is happening to proteins going into the membrane of the mitochondria. Again, post-translational. Let's see. Yep. And here we have vesicles, whatever. Okay, so now we're getting into Blobel and Doberstein stuff. So we've got ribosomes, and all RNAs start out being translated on free ribosomes. But if they happen to have a signal sequence at some point on their protein, then it gets bound by SRP. It ends up landing on the surface of the ER lumen, and it begins dumping the protein into the lumen, or interior of the ER. So, figure 15-14. This is a pretty nice figure to be able to work your way through and know what the different parts are doing here. We've got the signal sequence is bound by the SRP. The SRP, in turn, binds to the SRP receptor, and it now allows for the protein to, it halts the translation. Translation resumes, and the protein is being translocated across the membrane as it's being translated. Co-translational translocation. So, one last bit. When Doberstein was showing the boss that in the presence of the vesicles, the protein was at its original size, and in the absence of the vesicles, the protein was a little bit bigger. The reason is there is an enzyme known as signal peptidase that clips the signal sequence off of the protein. And if all you have is a signal sequence, the protein is going to get clipped and it will be floating around in the ER lumen. And next stop for this guy is going to be the Golgi apparatus, and then after that, it is secretory city. It's going to be dumped out. And so, elaborating on this theme, we can actually create membrane or transmembrane proteins, membrane-spanning proteins. And we'll pick up on that on Friday. Back on.